lines, ES, TS, and XEN cell lines. Similar to the mouse models, human blastoid models were created with various types of pluripotent stem cells that are believed to confer different pluripotency states. Almost all the findings in human blastoids are boosted in 2021, while some labs use human ESC and iPSC grown in naive conditions. Others have used EPSC conditions. Also, one group have used direct reprogramming of fibroblasts to form blastoids. This diversity in starting material likely contributes to the variation in blastoid generation efficiency and the capacity for lineage specification. Moreover, depending on the pluripotent state, triggers for directing the founding cells towards an organized blastocyst-like structure differs as well. As noted before, human pluripotent stem cells appear more readily able to contribute to TE lineage when they are cultured in naive or EPSC conditions than mouse pluripotent stem cells. This might explain why, contrary to marine blastoids, majority of human blastoids have been formed from a single cell type with high yields in just one single cell type approach. In different reported protocols, the blastoids are not uniform and also not consistent overall. When the blastoid formation are ready, how to evaluate them or compare the quality of different blastoids is another worthy considering question. The key is to see how similar they are between blastoids in vitro and blastocysts in vivo. In the structure, it's obvious to see whether the blastoids can form a cup-like egg cylinder in mice or biolaminar disc structure in humans. The expressing of different lineage markers should also be corresponded with different cell lineage to indicate the molecular and cellular events happened in vivo. Meantime, blastoid transcriptome profile by single-cell RNA-seq is also needed to inspect those of pre-implantation embryos globally in cell clusters and also developmental trajectory. Then, obviously, the formation efficiency is an important aspect limiting whether they can be applied on a large scale. Most importantly, the quality of the derived blastoid, especially the developmental potential when implanted in vivo, is responsible for investigating the cellular event's nature. Since the derived mouse and the human blastoids are already successfully, the next question is considering what we can do use blastoids. There is no doubt that blastoids are a valuable addition to research on early embryonic development. The post-implantation development happened in uterus that is very hard to observe. Blastoids can help us to uncover the developmental trajectory and milestones in early embryonic development, such as lineage segregation, implantation, and so on. Seeing is believing. Secondly, we can take blastoids as a useful tool to evaluate the pluripotency of our cultured or derived cell lines. Then, signaling pathways that are involved in blastocyst formation have been thoroughly studied over past decades in mouse. Based on these studies, we are now able to attribute the activity of several pathways to the development of the blastocyst lineages in both mouse and human. Consequently, small molecules that regulate these pathways are applied in blastoid generation. Lastly, the possibility of mimicking the pre-implantation embryo has huge implications for physiological studies, such as clinical studies, toxic screens, development of improved IVF compounds, and also studies on peri-implantation cell differentiation. Of course, the debate about how faithfully the in vitro derived blastoids can recapitulate all the features of the developing blastocysts is ongoing. In the future, the more perfect stem cell-based blastoid model and increasingly meaningful comparison can be made as these models to recapitulate early embryogenesis more faithfully. Comparisons between mice and human embryogenesis, these two model systems will continue to evolve alongside with each other and together have broad future applications, both for fundamental and clinical research. At the end of the talk, I'd like to introduce the research interests in our lab. Our goal is to dissect the regulation of pluripotent states in genetic, epigenetic, and post-transcriptional level by taking embryonic stem cells and primordial germ cells as the cell contexts. We have used a combination of functional genetics, genomics, cell biology, and developmental biology tools to systematically identify and dissect the function of regulators in pluripotent stem cells. If you have an interest in stem cell biology, please feel free to reach out to us. We are recruiting now. As for me, I focus on primordial germ cell specification, trying to answer how germ cells specified in vitro and in vivo. In mammals, the pluripotent epiblast in the post-implantation embryos gave rise to both the somatic and germ cell lineages during subsequent development. In response to signals from the surrounding cells, a small number of epiblast cells adopt the fate of the primordial germ cell and become the multipotent precursor of germ cells. Fortunately, the epiblast to primordial germ cell specification can be replicated in vitro using embryonic stem cells. However, the differentiation efficiency is not as high as we expected. So we want to employ two different strategies to investigate the germ cell differentiation process. One is that CRISPR-mediated genetic screening identifies key regulators responsible for germ cell specification. Another is that single omics technology profiles the process to describe the roadmap about why some input cells can differentiate into germ cells, others are not. I will stop here and thanks for your listening.